I'm Kevin Bender. Apparently, you've seen me already today, even though I haven't been in the room, because I have made fun of myself repeatedly, extensively, and that is my job. I'm a researcher over at UCSF, and we are primarily a little squirrely, furry mouse lab, but we've been sort of extending into other realms, and one of them has been working with all of you to do various assays that allow us to translate from what we know in, or what we could know in a mouse, where we can do a lot of experiments and ask a lot of interesting questions, to what we could potentially learn and extend over to kiddos. And that's where, that's one of the places where you guys have been so centrally important for what we do. So I just want to give you a quick update about what kind of things we're up to in our lab. We are asking questions about why some kiddos with loss of function get seizures while others do not, and we're asking that in mouse models and rat models. What kind of molecules, if you were able to design a molecule that sort of had the perfect function, what would it look like? How do those compare to what's currently being developed in the world, and how well could they work to suppress seizures in gain of function, misfunction, loss of function? When in life, and we're asking this in a mouse, could you see benefits from a rescue of channels? That's a centrally important question to everyone in this room. And then lastly, extending off of what Caitlin just talked about, what kind of assays can we use to tell if a treatment is working? And the real trick for us there is to ask a question of, if we're going to be testing these things in little rodents that are running around, that do their own little behaviors, that are completely different than what a kiddo can do, can we do something that translates between those two worlds? And so we've taken a view of going way back to the roots of evolution and studying things that are extremely simple, so simple that even a mouse could do it. And that's been our dogma. And so that led to this publication that was done with your help. Your kiddos are in this paper. If you've worked with us and done some eye tracking, you're going to see a few dots in a little bit. Those dots are your kids sometimes. This was all work done by Chen-Yu Wang, who recently graduated, Dr. Chen-Yu Wang, by the way. He was one of our first AP grant awardees. We just learned yesterday that Katie Salvati just got the AP award this year, so congratulations to her. And thank you to you for your support. And this is done in collaboration with Guy Bouvier, who is so cool, he only exists in black and white, and now has his own lab in France where he's continuing this sort of work as well. So what is this? I want to give you a really quick overview. So the impaired cerebellar plasticity, hypersensitizes sensory reflexes in SCN2A associated ASD, that's the paper. It is a page turner, I guarantee you. You can't put it down. If you want to read it, I'll print out a copy for you. But the long and the short of it is that we found, and Chen Yu and Guy found, that if you look in this little part of the back of your head called the cerebellum, and on the left you see the mouse version, that real high rainbow is SCN2A expression in cerebellum. You can see it's really high. And over on the other side is postmortem tissue from a human looking at NAV 1.2 or SCN2A in cerebellum. They actually look very similar. And so we were thinking that the roles for SCN2A and NAV 1.2 could be similar in these regions. I should mention that these things are extremely well conserved. The function of SCN2A and the function of what we studied can be found in primates like us, rodents. We could have done experiments in birds or crocodiles, but they bite too much. Can't get our hands on kangaroos that often. And so these are just things that all of us do, all the way back down to sharks. These are basically when you move your head in one direction, your eyes move in the other direction, whether you want to or not. This is something that you experience all throughout your life. And actually, one of the most robust ways of eliciting this experience is something we're all going to experience in 2 days. So if you go on the teacups and you go spinning around, think of me. It'll make me happy as you hear that sound, as you hear those noises. So unfortunately, we don't have teacups in the lab. What we have are little helmets and little chairs. And this is an experiment that started during the pandemic with making little helmets and having my own daughter be test kiddo number one. So Margaret was the first test on that helmet. And then you guys have helped. You can see us in the middle doing some tests last year in Boston. Where the helmet's gotten a little smaller. It's gotten smaller still, and then we've got this new version that allows us to do a few other fancy things, hopefully maybe even with EEG at the same time. But the long and the short of it is that when we move back and forth with the lights off, we measure your eyes moving in the opposite direction. And what we're looking for is a difference between neurotypical kids and kiddos with SCN2A loss of function. And the long and the short of this is that if you look in the top, you'll see what we got out of the kiddos, out of all of your kiddos. Neurotypical, the gray line is a little low. SCN2A loss of function, it's pretty high. The thing that's important here is that if you compare that to what we get in a mouse that has the same sort of genetic condition, we see things that are very, very similar. Okay? Almost like this is honestly the strongest mouse model behavior that we've ever seen and probably is present in the autism field right now. And so it's really, really useful, and we want to leverage this as much as possible. And so one way in which we can leverage this is we can ask, how well can we change these sort of behaviors? And so the way we do that is we let the mice go in their own version of the teacup ride, which looks like this. So the mouse sits in the middle, and what you'll see is that the mouse is on a platform like a teacup, and they're moving back and forth, and the whole world is moving back and forth too. And then we have a picture that takes a picture of their eyes. We do recordings from their brain at the same time. We will not do this with your kids, just so you know. But that allows us to get information about how we can change, how we can modify these sort of activities that occur in your brain and see whether or not they can be rescued or recovered in response to treatments. And so that's been one of the things. I'll skip through this real quick. This is what a normal animal looks like before and after going on teacups. Black is before, gray is after. You can see things have changed because we just induced a change. We don't see that in SCN2A kiddos. They stay pretty stable in what their responses are. And this is something that stays stable throughout life. But what we wanted to know is, yes, it's stable. Yes, it's not moving. But if we give some sort of treatment that in a mouse restores SCN2A, can we see plasticity? Can we see malleability in these sort of activities? And so we've been working with labs at UCSF, and this is stuff pioneered by Navneet Matharu, who's in the audience somewhere, and Perry Spratt. They've now working with us at Regal Therapeutics to develop approaches to do a CRISPR-based rescue of SCN2A. And the long and the short of it is, we know we have one good copy and one potentially bad copy. If we can tell the good copy to work harder, we might be able to get back to normal expression levels. And so we've been using these mice to test whether or not we can see that. And so here what you see are our normal case in black, our SCN2A case in cyan, and then below that is an SCN2A animal that was given this therapeutic at something called postnatal day 30. That's equivalent to about a 14-year-old kiddo. Okay, and at that age, if we give them that therapeutic, what you see is that we get a change, we get a rescue of plasticity, but we don't get a recovery from certain aspects. So it suggests that we can recover some aspects of learning, but the full rescue is not observed there. So that tells us something about what happens when we do it into an equivalent of a 14-year-old. But if we do it at postnatal day 3, which is extremely young for a mouse, couple years of life or earlier, we see a full rescue of these kind of things. And it's mapping at the level of individual animals. When we have success or failure in delivering this therapeutic, we can see success or failure. So it's something that's really, really quantitative for us, and it's extremely useful to be able to test a whole bevy of different approaches. And we now know also that this is translatable into kiddos. So this is something where if we can measure a kiddo, and we're starting to do this longitudinally, if we can measure before and after a future therapeutic, we might be able to leverage this as a way of saying, in an unbiased way, whether or not a therapeutic is working. I would imagine that we're going to see a whole bunch of other things that are going to be extremely biased that'll tell us that whether or not things are working. but this is another thing that we would need. Okay, so moving forward, there is this question, and this is sort of a big question, and one of the things that I want to thank you guys all for leading the field on. SCN2A is one of these major genes associated with autism spectrum disorder and intellectual disability. This is a figure from Stefan's lab showing just a distribution of a bunch of letters and words and numbers of all these different genes. What you'll notice there is SCN2A is way at the top. It's way over on the left. It's the most important gene in autism spectrum disorder, but there's a bunch of other genes. And one of the big questions we have is, now that we've learned this about this one gene and how robust this sort of behavior is in these reflexive eye movements, can that tell us about other genes on this list? And can we then dive deeper and develop better biomarkers for autism spectrum disorder in general? And so one of the ones I wanted to highlight here is this gene called ankyrin-2, which Paul Jenkins has been working very heavily on in his lab. It is the scaffold that allows NAV1.2 to sit in the membrane of a neuron and be in the right place. It turns out that if you look at, these are just pretty pictures, but I just want you to notice that they look very similar across them. One of them is a stain for NAV1.2 or SCN2A. The other one is a stain for ankyrin-2, or proteins called ankyrin B. They look very, very similar in their distribution in the cerebellum, suggesting that they might have similar mechanisms. And Amanda Stark in our lab has started doing experiments on these animals and are seeing some rather remarkable similarities between an animal that's an SCN2A loss of function and an animal that's an ankyrin-2 loss of function. And so this convergence across autism is something that we would only have learned if only for this group of people. So thank you very much for not only helping your kiddos, but basically starting to help a very large population of kiddos that don't even know it. So they'll learn. Okay, the other thing I want to say is just thank you for the support, and thank you for the help, and thank you for pushing us. I was talking with Karen earlier today about just like, why are we doing this? How the heck do we do this? We're a mouse lab, and I would only do this because I've come to these meetings, I've met you guys, and I thought, if there's a group of people that could help me go along this way and help us go along this way, it's you. So thank you for participating in all this. I look forward to participating with a bunch of other people. Basically, any kiddo that's tired of sibling camp, send them over, and we'll have them do something even more boring. But Chen Yu did this wonderful work as an AP awardee through your direct support. Guy now has his own lab in France. He just was awarded the major European award for young scientists. It's called the European Research Council Award because of this work. So this work allowed his lab to grow in this massive way. And now this question about what is going on with autism in general, we're doing now in collaboration with Jason Christie over at Colorado and Summer Bishop at UCSF. We're cerebellum experts, child psychiatrists and psychologists. We're bringing in as many people from the local community as possible to do these sort of things and ask, how far can we run with these sort of ideas? Lastly, I want to thank everybody in my lab and a lot of whom are here who would love to chat with you. I will mention that there was another thing that came up already, puberty. We're starting to think about that directly in terms of relationship to the estrus cycle. So Barbara's doing a lot of this. So anyone who has a kiddo who's going through puberty, Barbara would love to chat with you. And just thank you to you guys and all the collaborators that are associated with us.